veil because it's kind of hard to pull apart. What we see is we see our drug binding to our receptor, and then we see our response, and then there's all this stuff that goes on behind the veil in between. And so we can see very different responses from the same stimulus depending on the cellular cascade, the abilities of that cell in order to respond to that stimulus. So that's what we're seeing in this figure here. All the way on the right, we see 4 different sets of graphs. Presumably each line on the graph represents a different drug, and each graph represents a different cell system. And so we see that the drugs behave very, very differently in each system. When we talk about cellular cascades, usually it is going to be an amplification system. And this comes back to a point that we made, I think, either in the last video or the one before that, about not needing all of the receptors. Remember when we talked about efficacy-driven or affinity-driven agonists, we talked about the effect of having more receptors than you needed. Where with an efficacy-based agonist, we didn't have to occupy all of the receptors in order to achieve 100% response. This brings us to the same idea, but we're going to give it a special term, and that is receptor reserve or the reserve receptors. The receptor reserve is how many extra receptors we have once we have obtained 100% response. So in our graph here, on the x-axis, we have percent occupancy. It doesn't go up to 100%. We don't need it to. We're presumed in at the very beginning of our graph, and the y-axis is just our percent response. And so we see 3 different agonists of the same receptor, each have slightly different occupancies. Each have slightly different response rates at the same occupancy percentage. So isoproterenol achieves 100% response at occupancy so low that we can't even tell that it's not at 0. Basically, any occupancy with isoproterenol achieves 100% response. This BRL compound achieves 100% response at very low occupancy. It is certainly more than isoproterenol, but at just 10% occupancy, we're at essentially 100% response. Whereas the CGP compound requires a lot of occupancy to achieve response. And so at 10% occupancy, we're at roughly 50% response. And so isoproterenol has a much larger receptor reserve than CGP. For isoproterenol, roughly 99% of the receptors, or even more than 99.9% of the receptors, are extra. They're unneeded, and so they are the receptor reserve. Another thing that influences the response due to cellular mechanisms are internal mechanisms. And so this figure is showing the response with G proteins and G protein coupled receptors. And so we've already talked about this, but in this situation, all we really talked about was step 1, the receptor becoming activated, and a little bit about step 2, where the G protein becomes activated. That is more or less a ligand receptor process. However, what's actually going to happen due to the activation of the G protein coupled receptor and the G protein itself is going to be downstream of multiple processes. So once that G protein becomes activated, the G protein is actually made up of multiple protein subunits. And so it has to separate. Those subunits have to move somewhere else in the cell. They have to bind to and exert some sort of influence on another protein. In some situations, it's going to activate that other protein. In some situations, it's going to inhibit it. Once that is activated, the G protein has to become inactive again in order to be recycled. So it does that by breaking down GTP into GDP. A G protein that is bound to GDP is inactive. Subunits now have to come back together again, and now we're on step 6. Once we're at that point, now it can come back up, bind another activated G protein coupled receptor, and do the whole thing over again. But you can see that these processes involve a lot of things other than the ligand, the receptor, and the G protein. We have to have GTP available for the G protein to become activated. We have to have the affected proteins available. The GTP has to be hydrolyzed, and then the subunits all have to find each other again to come together. If we are working in 2 different systems, those mechanisms are going to work differently in those 2 systems. And because of that, the response to our ligand binding our G protein coupled receptor is going to be different in different systems. This is another example of a signal cascade. I'm not going to walk you through it. It's not important. I don't need you to know that G protein coupled receptor system by heart. I just need you to understand how individual portions of an intracellular cascade can influence the response that we receive due to a ligand binding to our receptor. Like I mentioned, usually this cascade is an amplification system. So that's what we're looking at here. The x and y axis, they have no units. These are not meaningful things. This is just a mathematical equation. This is not a scientific idea. What we see, just imagine this is the value of x and y is a function of x. And so if we put the value of x into a function and we get the value of y, we get this function 1 curve. Now, if we come and we say that z is a function of y and y is a function of x, essentially what we've done is we've nested 2 functions together. So that is this one. function 1 plus function 2. And so essentially what's happening is function 2 multiplies function 1. So imagine we take this graph, and whatever value we get, we multiply by 2. I don't know if that's the number here, but you can imagine you're going to amplify and magnify that curve, and it's going to look something like this. This is a figure that is very similar to one that we saw in the occupancy theory lecture, where we have graphs representing a stimulus at the beginning, response at the end, and then a function that converts stimulus into response in the middle. So here at the beginning, we have two different agonists. Both have very different stimulus curves. As we go up in agonist concentration, agonist 1 does not achieve nearly the amount of stimulus as agonist 2, and it looks like maybe the EC50s are different. It looks like it probably is different, but it's not incredibly obvious to the naked eye. If we take these functions and then plug them into the stimulus response cascade function, we get this, where agonist 1 and agonist 2 both achieve 100% response. The only difference between them is the EC50 or the potency. And so for stimulus, they had different efficacies. However, in terms of response, they did not. The amount of amplification that we see is very much dependent on the system that we are working on. There are two graphs here. They're quite complicated in terms of details, but I don't need you to know the details. I need you to know the gist of them and understand what we're getting at. In this situation, on the left, x-axis is just showing occupancy, y-axis is response. In this situation, we are giving an agonist, the same agonist, to the same receptor in 3 different types of cells and measuring the response in each one. And so we can see that in 3 different types of cells, melanophores, CHO cells, which are Chinese hamster ovary cells, and HEK cells, which are human embryonic kidney cells, we get very much different responses. We see the same thing in tissues. On the right, we're adding increasing amounts of the agonist prenalterol, and the y-axis is % response. We apply the same agonist, same concentration of agonist, to tissues that contain the same receptor, and we see very different responses depending on which portion of a heart that we apply that agonist to. We see for this tissue, we get almost no response, whereas in this tissue, in the right atria, we achieve essentially 100% response. We can also see differences in amplification depending on what we are measuring as our response. Usually, the further we get from our stimulus down a signal cascade, the more the amplification is going to occur. If we measure our response 3 steps down the cascade from our stimulus, we're going to see less amplification than if we measure something that is 10 steps down the stimulus. And we can imagine this as a lever balancing on a fulcrum. So in this situation, we have a plate hanging from one end, and we add an agonist to that plate. And the weight of that agonist is going to be imagined as our efficacy. The more efficacious it is, the more it's going to pull down on the left end of our fulcrum here, which brings our right end higher up. And you can see that the further we get from the fulcrum point, the higher up we get. So in this situation, we have a threshold that if our response is below this threshold, we cannot measure it. And potentially there is no response at all. We also have a maximum response. This is the 100% response, and we cannot go above that. So in this situation, if we measure this far away from the stimulus, we see a small amount of response. And the further we get, our response curve gets larger and larger until we get far enough away that we hit the 100% response line. Now this can be complicated because this amplification can hide differences in efficacy between 2 compounds. So here we have 2 different levers, and each one has a different agonist, agonist 1 and agonist 2, and we can see that agonist 2 is more efficacious than agonist 1. Agonist 2 is heavier than agonist 1, and so it causes the lever to go higher. However, if we look so far down the cascade that both of them have reached the 100% response level, in that situation, we are not going to see differences in efficacy between them. That is the third graph here. Agonist 1 and agonist 2 both reach 100% response, but they have different potencies. They have different EC50s. Below that, though, at this medium level, agonist 2 has reached 100% response, agonist 1 has not, and so we can see the efficacy differences. We can also see them very early on at this first graph, although neither one of them has reached 100% response. And so you can imagine that picking your experimental conditions and where you're measuring your response is going to have a huge impact on whether you can see efficacy differences between 2 compounds and whether you see something as a partial agonist or a full agonist. To make this even more complicated, one stimulus can have multiple efficacies. Signal cascades can split and branch. And so a stimulus activates one protein. This one protein activates 2 different proteins, kicking off 2 different signal cascades that have very different responses and different efficacies. So in this situation, we have added cyclic AMP to some tissue, and we measure 2 different responses. One of them is force of contraction. One is rate of relaxation of atria of a heart. And so we can see that there are very different response curves for the different responses. But they both came from the same stimulus, the cyclic AMP binding to a single receptor. We can tweak this. We can do it purposely, but we also have to be aware of tweaking that is occurring unintentionally that is going to alter our results. So we have two separate graphs here, two separate situations showing two different examples of how this can happen. On the left, we are looking at the effect of phosphodiesterase enzyme. So phosphodiesterase degrades cyclic AMP. And so as we add cyclic AMP, we are going to achieve a response. Cyclic AMP is our agonist here. And so the line dark circles is our control experiment. As we activate receptors by adding cyclic AMP, we get a certain amount of response. However, let me back up. Cyclic AMP is not our agonist. We are adding a different agonist, isoproterenol. Isoproterenol binds our receptor and kicks off a signal transduction cascade that involves cyclic AMP. And so, in both situations, with and without phosphodiesterase, we would achieve the same receptor activation at the same concentration of isoproterenol. However, with the white circles, phosphodiesterase is around, degrading the cyclic AMP. So the same receptor activation, the same stimulus, achieves different responses because with the white circles, the response cascade is inhibited through the degradation of cyclic AMP. On the right, our agonist is carbachol, and as we add increasing amounts of carbachol to our receptor, we get the same amount of stimulus again. However, in this situation, our response that we're measuring is muscle contraction. And the process of muscle contraction requires calcium ions. So in the presence of large amounts of calcium ions, we get the black curve. But in the absence of, or very low concentrations of calcium, we get the white curve. And so we can see that the available calcium in our cell is going to alter the apparent efficacy of our agonist. Something else to keep in mind, I think this is the last slide, Last slide is receptor desensitization. Receptor desensitization is something I think we're going to talk about a little bit more later on, but this is partially a cellular process. There are multiple mechanisms through which receptors can be desensitized. In some situations, it is a cellular process, meaning it's going to be system dependent. And so on the left graph, we have increasing amounts of isoproterenol, and when we add increasing amounts of isoproterenol, we get the black dot curve. If we incubate the same cells with isoproterenol for 90 minutes, withdraw or wash out the isoproterenol, and then add it again, we get a different curve, the white square curve. We have shifted to the right. We have apparently decreased the potency of isoproterenol. But note that they have both, in both curves, reached 100% response. In the same system, with a different agonist, pernalterol, doing the same thing, the first time we add the agonist, we get the black curve. On the second time around, after 90% or 90 minutes of pernalterol activation, we wash it out, we give it again, and we get essentially no activation. In this situation, receptor desensitization inhibited efficacy, whereas over here, it inhibited potency. So receptor desensitization is a complicated process. I think we'll talk about it more later on, but here you need to realize that it can be a cellular process, and it can inhibit potency and or efficacy.